So thank you, Corey, for inviting me, and thanks everyone for showing up. I realize the sort of data that we do in my lab is probably a long way from what most people do. So what I tend to do for a broader audience is really to focus on mostly zoological questions, and that really is what drives interest in my lab. We use developmental biology as a set of tools to address basic zoological hypotheses. And while most people focus on adults, our lab uses the early developmental stages and larval stages to address basic hypotheses. So when we think of an asteroid or a sea cucumber, as shown here, we generally think of them as they are adult. But there's this magical long period in the plankton that no one really thinks about, where the body plan of the adult looks very different from the body plan of the larva. And so my lab is really interested in what information we can get in terms of understanding zoology and the diversification of animal phyla by looking at the early developmental time points and the development of larvae. So the primary research interests in the lab are evolution of developmental mechanisms. So if you want to understand the final morphology of adults, you actually have to understand how the adult gets put together. And it's the process of development that actually assembles the animal into their major constituents. And so my lab is really interested in how you actually piece together an animal body plan and how that changes over evolutionary time periods. So how developmental biology changes defines the major divisions of our body. The antero-posterior axis, the dorsoventral axis, the left-right axis, all of that is defined during the developmental time period. And so it's that macroevolution of animal body plans that I'm interested in. How did animal phyla diverge, and how did the major body plans of those phyla get defined, and how do they differ, and what does developmental biology tell us about those differences? And so what role can developmental genetic data play in reconstructing the early evolution of animal body plans? So we're used to thinking about reconstructing the evolution of animal phyla by using morphology. Comparative anatomy has played an enormous role in understanding the evolution of different animal phyla. We're interested in the developmental mechanism that actually sets up those major anatomical features that classically have been compared across different phyla. And we particularly have used new perspectives from unusual or / understudied phyla. So those of you who have taken your basic developmental biology courses will know that typically there's mainly two phyla that people focus on in terms of developmental biology. Chordates, which includes mouse and zebrafish, and chick, a variety of vertebrate model systems. And then arthropods. So Drosophila melanogaster is one of the most powerful and important developmental model systems. However, when you start moving outside of the arthropods and chordates, it gets pretty thin on the ground. So if the goal, one of my goals, is to try and understand the origin and diversification of phyla, making a bipartite comparison between two different phyla with very unusual morphologies to reconstruct to an early ancestor is problematic. You need to sample a more diverse array of animals with more diverse anatomies. And so we typically have studied animals that generally people haven't paid much attention to or have a highly unusual body plan in the hopes that these outliers will tell us a little bit more about generalities about the evolution of body plans. So there's a Nature paper every year that basically reorganizes the topology of the animal tree of life, but this is one from a few years ago, which basically shows that if you're interested in the origin of bilaterians being animals that all have an antero-posterior axis, a dorsoventral axis, and a left-right axis. Most of the animals that we're familiar with, except for cnidarians, ctenophores, and sponges. So if you're interested in the origin of bilaterians, then most people are focused on the question of whether or not they're complex or simple. And classical zoologists would argue that the earliest bilaterians were probably pretty simple animals that secondarily evolved more complex body plans in animals like chordates and arthropods. And this has been generally backed up by the fossil record. So most of what we understand about the early origin of life comes from a lot of paleontology that was done on a period of time in the Cambrian. So during this period of time between 545 million years to 450 million years, that is when most of the animal phyla appear in the fossil record, with unambiguous fossils which tell us that that was the early origin of those groups of animals. So if you follow the graph along the x-axis, you can see that at the base of the Cambrian, you start to see evidence of different phyla appearing. So phyla are in blue. Actually, I've got my little... So here you can see that at the base of the Cambrian, we start to see a burst in the appearance of phyla, and then that stabilizes by the middle of the Cambrian, and basically there's no more phyla. The classes also start to increase in number during the Cambrian and increase in size as you get to more modern geological time periods. But basically, there's very little in the way of animal diversity below the Cambrian. There's this group of animals called the Ediacarans that no one really knows what to do with. But basically, 545 million years ago was when most of the animal phyla appeared in the fossil record. However, if you look at these lines here, these are actually molecular clock predictions about when the animal phyla diverged. And so if you look down here, this is the projected time period where there was divergence and the origin of bilaterians. So this sort of mythical animal that people are interested in working out that formed all of the bilaterians, some people predict appeared around about 700 million years ago. This moves up and down depending on the analysis, but most people are in agreement that the molecular clock predictions really predate the fossil record by quite a substantial period, suggesting that there's a lot of cryptic evolution of bilaterians that happened long before the Cambrian explosion, suggesting we know very little about the early origins of the major phyla because there's no impression in their fossil record. And this generally, so this long cryptic period of bilaterians are generally interpreted by paleontologists suggests that the earliest bilaterians were likely epibenthic grazers, deposit feeders, and they were likely to be very small and simple, which is why they didn't fossilize very well and why there's very little evidence for complex bilaterians before the Cambrian. However, this has been challenged by molecular genetics, and so over the past 30 or 40 years, we've got to know an awful lot about the basic molecular biology of how you set up the animal body plan of animals. And one of the most surprising findings started off in Drosophila and was then followed up on in chordates, was that if you look at how we set up our basic nervous system on our basic anteroposterior axis, so anterior to posterior, these color coding of regions of the brain of sort of idealized chordate here, and then a Drosophila melanogaster here. So if you look at the protocerebrum, which is the anterior brain, the deuterocerebrum, the sort of middle brain area, and then sort of high brain and spinal cord and nerve cord region, the genes that turn on the programs that define the anterior brain of Drosophila are the exact same genes that are involved in turning on the forebrain program in a chordate. Similarly, genes that turn on the sort of middle brain region of Drosophila are the same genes that turn on the program of the midbrain, hindbrain, and the spinal cord, a very similar program of genes that turn on the nerve cord in arthropods. And some of you may have heard a group of genes called the Hox genes, which is a very unusual group of genes because they're arranged in chromosomes in a 5' to 3' order. So Hox 1, 2, 3, 4, 5, 6, 7 are organized in their expression domains, match their actual arrangement on the chromosome. So the most 5' of the Hox genes are expressed in the most anterior domain. As you go down in the position in the chromosome, they're expressed in an increasingly posterior domain. So organization of the chromosome matches actual expression in the body plan of the animal. And they found that in Drosophila and in chordates, that the Hox complex and their expression of genes in sort of lockstep down the body plan are basically the same. So people have always really focused on this role in the central nervous system. So basically the central nervous system of chordates and Drosophila and how they're set up, where they're positioned on the dorsoventral axis, and how they're organized along the AP axis. The same suite of genes defines very similar domains in both of those groups. So this has led to the argument by a lot of people, and these are not shabby reviews. This was published in Cell back in 2009. There's quite a collection of these kinds of reviews that have argued that based on this molecular genetic data, that it's almost impossible to conclude anything other than the earliest bilaterians were actually pretty complicated little beasts. That they weren't these really simple animals with probably more like a nerve net. They actually probably had a central nervous system. So what I've always argued that these reconstructions really assume that we understand the relationship between these conserved gene regulatory networks and the evolution of morphology over large time periods. And typically, as I said, developmental biology tends to focus on certain groups of animals. So obviously, Drosophila melanogaster, the mouse here. There's now a new animal from the larger group of animals, bilaterians of the Lowe trochosomes. This is a polychaete annelid. That again, all of these animals share very similar types of body plans. They have a highly complex sensory system, anterior sensory system. All of them have eyes. All of them have pretty complicated sensory systems in the anterior. And then they also have a very highly complex central nervous system and complex motor outputs. So they're often predatory, and they're often highly mobile. And the question is, does this really represent what... It's the kind of animals we need to represent the early evolution of life. So if we look at another Deuterostome that is in the same main lineage of animals as mouse, then we have Hemichordates, which have very little in the way of a central nervous system. If you look at other annelids, you look at Ezechiurin, other annelids look completely different to what we see in these sort of highly complex central nervous systems. And then if you look at other ecdysozoans that are in the same clade as Drosophila, then you can see this parapelid here with very little of the kinds of specializations that have been used to study and reconstruct early evolution of animals. So the question that we're interested in is, what can these more modest animals tell us about the relationship between body plan evolution and conserved molecular genetic networks? So to understand the relationship of these gene regulatory networks with the evolution of animal body plans, we need to sample a better sampling of body plan diversity. And so we selected a group of animals called Hemichordates as an example of a simple body plan that would contrast with the studies of more complex developmental model species. So I'm particularly interested in the group of animals called the Deuterostomes, which is the major lineage of animals that our phylum belongs to. So we have chordates here in the Deuterostomes, which is composed of the vertebrates, tunicates, and Amphioxus, which is a very simple but very basic-looking chordate. And then Hemichordates are the sister group to the Echinoderms, and then they're more distantly related to the chordates. So I actually started my postdoc. I started my PhD work on Echinoderms, and through that work, I started to get interested in Hemichordates. And on reading on Hemichordates, the big monographs of Hemichordate development and Hemichordate biology were back in the 1890s and early 1900s, and William Bateson and Thomas Hunt Morgan worked on these animals, but suddenly the work on Hemichordates after the 1920s and 30s basically disappeared, and there were a handful of papers, but very little work was done on them. And so naively, I decided that I thought I would do my postdoc on Hemichordate development because I couldn't believe that no one had seen what a great group of animals this was to work on. And then I spent the next year trying to find a Hemichordate, which then made me realize that actually there's a good reason why people haven't worked on Hemichordates before. So I had a lot of fun traveling around trying to find a species to work on. So I went to a lot of marine stations in the tropics trying to find a Hemichordate, and I actually spent a pretty shameful 2 days digging out what I thought was a hemichordate and then realized it was actually an echiurine, so I spent 2 days digging out echiurines rather than hemichordate. And so finally, after a year, I settled on a group of hemichordates that have been described at the Marine Biological Laboratory at Woods Hole. So quite a lot of work had been done on them in the 1950s, and the basic reproduction had generally been already solved. And that, for the kinds of work I do, is part and parcel of the main problem, is the basic biology of many of these groups is really poorly described. And so the molecular biology itself is actually pretty straightforward, because DNA and RNA are DNA and RNA, no matter where you get them from. But the main challenge is actually getting the animals to reproduce and to work with the embryos. And that's where a lot of the work that is never published, because no one really cares about it, but often that is the challenge of doing this work, is actually getting the animals to reproduce and to work with the embryos. So a big question in Deuterostomes is, how do you even make even basic comparisons across the body plans? And I think this is a really, if you get only one thing out of the talk, this sort of main point, I think, is an important one. Because we're so fascinated by vertebrates, we generally ignore the body plans of other Deuterostomes. And so if we want to understand the early origin of Deuterostomes, then we're left with some pretty challenging body plans to work with. So how do you make even a basic comparison between the hemichordate and a vertebrate? Where is the head in the hemichordate? How does that compare to the anterior region of the brain of a vertebrate? Even more challenging is the asteroids, or the echinoderms. I would challenge anyone to tell me where is the head in an echinoderm. Even after many years of zoology, people still don't even know where the basic anterior of an echinoderm is. There's no data, there's no morphology that really helps you understand that kind of comparison. So this particular reconstruction is really problematic. With the range of morphologies we have are just too diverse, and the fossil record is too poor for us to make any conclusions about what this early Deuterostome might have looked like. So what we're really interested in is, given that we know so much about the molecular genetics as a kind of a second anatomy, as a kind of a hidden anatomy that's built under the skin in animals, how can we use this hidden anatomy in these different groups to reconstruct the early evolution of the Deuterostomes? So this is the beast that we have done a lot of work on. It's called Zachyglossus kowalewskii, and the animals are about that big when you dig them out of the ground. They're very negatively phototactic. They start digging as soon as you pull them out. They have this anterior proboscis that it uses for digging and feeding, which give way to a ventral mouth between the collar and the proboscis. And then there's paired gill slits, which you can see the gill slits here in this panel. And the animal feeds largely by deposit feeding. Here you can see the anterior proboscis pushing back particles trapped in mucus back towards the mouth. But then they also wave that proboscis around, and then they can directly filter feed, and then the water goes into the anterior part of the gut and then is passed through the gills. These gills, based on our work, look completely similar, both morphologically and molecularly, to the gills of chordates, hence one of the names Hemichordates. They are different sexes, so you can sex the female and the male very easily just by the color of the gonad. So the female produces these sort of greenish eggs, and the male has a much more sort of milky color gonad. They live anywhere from intertidal to deep waters. Interestingly, they're very hard to find in California because they generally live in inlets, and there's very few nice inlets in California. Most of it's is open rocky coastline. It's too muddy here, so I've actually been digging around in this area trying to find them, but I've never found them in this area. The only place I've actually found them now, my graduate student actually found them down in Morro Bay, but we haven't found any of them up and down the coastline here, so they're actually difficult to find in California. So in terms of their body plan organization, this is the anterior proboscis, which is used for the digging and feeding. This is where the mouth opens up on the ventral side, passes through into the anterior gut. These are the gill slits, which then allow the animal to filter feed. And then they have some pretty interesting elements to their nervous system. They have two cords. They have a dorsal cord and a ventral cord, but then a very broadly organized nerve net. So here's a little juvenile. This is the proboscis, collar, and trunk. And there's a stain that shows basically all of the neural processes. And you can see here there's a very broad net of a meshwork of axons in the proboscis and in the collar, and then it becomes less in the trunk. But then you can see a dorsal and a ventral cord running down both midlines. So it has both these centralized elements in the cords, but it also has this really pervasive nerve net, which is quite different to what we see in invertebrates. So its nervous system is organized in a way that's it's a striking contrast to what we normally look at in terms of model species. It doesn't have the level of centralization that we're used to seeing in Drosophila or in the chordate. So, one of the major questions is how flexible is the role of these conserved molecular networks in patterning diverse body plans? So, we know that these genes are present in this animal. The question is, what are they doing in an animal that has such a contrasting nervous system organization? And some of these may get a little on the, maybe more than people really want to think about, but I'll always emphasize the parts that I think are most important. So are these transcriptional networks, which are conserved between vertebrates and arthropods, are they coupled to specific morphological structures at evolutionary time periods? So given that people have generally used it to reconstruct a complex nervous system, are they always associated with a complex nervous system so they won't be expressed in a compelling way in a hemichordate? Or are they much more flexible? So are they much more malleable in the way that they've been used in different types of body plans? And so finally, the question which I've gotten a lot of flack for over the years is can comparative molecular genetic data be used to reliably reconstruct ancestral morphology? So is it a useful data set for reconstructing ancestral morphologies? And then finally, back to that question where I showed you the 3 different body plans in the Deuterostomes, can molecular genetic data facilitate body plan comparisons between animals with very divergent morphologies? So just a very basic intro to the early development of Saccoglossus. So the animal goes through basic cleavages after fertilization. So it blows up this big vitelline envelope that protects the embryo. Then it starts to go through cleavage stages in a very equal divisions. And then it eventually will form a hollow blastula, which is basically a hollow ball of cells. And the major axis that forms this is what's called an animal-vegetal axis. And this is very common in embryos. All the region in the animal pole that's coded blue will give the ectoderm. And in the vegetal pole, the spotted yellow and red is all of the tissue that will give rise to the endoderm and the mesoderm, the muscles and the gut. So at gastrulation, the embryo folds inwards. So this vegetal pole folds inwards and touches the animal pole. So you get the internalization of all the endoderm and the mesoderm. And where the vegetal pole touches the animal pole, this region here will be the entire proboscis. So you start to get a little divot in the ectoderm of the embryo. So this little boundary here shows what will become the proboscis ectoderm and the proboscis mesoderm. So all the muscle of the proboscis comes from this region of the embryo. All of the ectoderm of the proboscis comes from this region of the embryo. You then get another little divot in the ectoderm, and that is the boundary between the collar and the trunk. And then this little region of mesoderm will go on and form all the muscles of the collar. And then finally, you get the pouching of the final part of the coeloms, which will give rise to all of the mesoderm of the trunk. So it's a very delineated body plan. It's very easy to understand. This is what the embryos look like after they've gastrulated. So we're now looking at this stage. So they swim around in the vitelline. And then you can see, even by day 3, these animals are actually baby worms. So you have a proboscis, a collar, and a trunk. Proboscis, collar, and trunk. Here you can clearly see the proboscis, collar, and trunk, and they still are encapsulated in the vitelline envelope. Here you can see at day 5, they already have their little gill slits. So one pair of gill slits are formed. So at this stage, they're basically baby adults ready to hatch and to start digging and feeding. And so this is after hatching, they swim around, and then by about day 10 of development after they've been feeding, they start to elongate the trunk, and they're forming little burrows. So what have we done in terms of developing molecular infrastructure? So we can do what's called in situ hybridization, which many of you heard of. It's a way, as is shown in this embryo, of labeling up where certain genes are expressed in certain cells. So I'll be showing a lot of in situ data. So basically, wherever you see a dye precipitated in the embryo, that's a way that we can determine where a specific gene is expressed. So we've cloned lots of different genes by sequencing RNA from a variety of different developmental stages, giving these comprehensive EST collections. So we have hundreds and thousands of genes we've cloned using screens of EST collections. We recently published the genome for this animal about 2 or 3 months ago. And then we can actually manipulate gene function. So we can actually knock down gene function by injecting interfering RNAs. And we can also manipulate gene function by overexpressing a gene, seeing what happens when you inject too much of the gene in an embryo. And then we're just starting to develop transgenic approaches as well. So these data show you the kinds of data that we use to compare the hemichordate data to. So this actually shows you that suite of genes which are involved in specifying the major regions of the vertebrate central nervous system. So I'm going to show you a couple of these genes. So this gene is called 6'3". The names are not important. It's really the pattern that I want you to pay attention to. So here's a little mouse embryo showing this is the anterior brain, this is the midbrain, this is the hindbrain and the spinal cord wrapping around. And you can see wherever the purple dye is precipitated, that's where the gene's expressed. So this gene 6'3" is expressed in the forebrain. This gene NK2.1, again, is expressed in the forebrain, but more ventrally in the forebrain, and also in the limb buds. This gene called OTX is expressed a little bit in the forebrain, but mainly now into the midbrain region, all the way down to the boundary of the hindbrain. And another gene engrailed is expressed at the boundary between the midbrain and the hindbrain. And then finally, those Hox genes I was telling you about that have the same spatial expression down the axis as their organization in the chromosome, these genes are markers of the central nervous system and also of the mesoderm. So HOX1 is expressed in the most anterior domain, HOX5 in the sort of more medial domain, and HOX9 in the most posterior domain of the developing central nervous system. So we cloned many of these genes. So I'm showing you a subset, but we've got data from 50 to 100 of these genes that are involved in marking defined domains of the central nervous system. So if you now look at these in hemichordates, so if we look at this gene, both of these genes are forebrain genes, both in Drosophila and in vertebrates. And in hemichordate embryos, they're expressed exclusively in the proboscis ectoderm. So here's the proboscis, collar, and trunk. Proboscis, collar, and trunk. And so both of these genes that are markers of forebrain are also expressed in the hemichordate. However, they're not expressed uniquely on the dorsal, the ventral midline, which is where a CNS might be. They're expressed in rings around the entire ectoderm, which is where the basiepithelial nerve net is. So it's not marking necessarily specifically a central nervous system, it's actually marking nervous system in general. But if you now look at genes that are expressed a bit more posteriorly in the vertebrate brain, we also see in hemichordates they're expressed more posteriorly. So this gene OTX, which is expressed in the midbrain all the way down to the boundary of the hindbrain, you can see here is expressed at the base of the proboscis into the collar and anterior trunk. Engrailed, which is a really good marker of the boundary between the midbrain and the hindbrain, is at the boundary between the anterior trunk and the collar. And then the HOX genes are expressed, HOX1 is expressed in the anterior trunk overlapping with engrailed, and then HOX5 is expressed more broadly in the trunk, and then finally HOX910 at the very posterior trunk. So the general trajectory, the actual relative position of these genes down the axis is identical between these two groups. how they're organized along the dorsoventral axis reflects the organization of the nervous system. But they aren't marking anything near similar in terms of the morphology of the nervous system. They're completely different in their organization. So what we concluded from this work, so if you look at, so this is data summarized for about 30 different genes when we published back in 2003. So basically, genes involved in forebrain formation are expressed broadly in the proboscis. Genes involved in midbrain formation are expressed broadly in the collar. And then hindbrain and spinal cord genes are expressed broadly in the trunk. So there's these similar expression topologies for many of these important transcription factors involved in central nervous system AP patterning, despite this enormous morphological disparity in body plan organization. So clearly there's broad expression in the hemichordate ectoderm, which suggests that over macroevolutionary time frames, these genes are much more generic in their patterning role. They don't mark specific morphological neural structures. They actually seem to provide pattern for specifying a range of different neural topologies. So we've argued that this suggests a much more complex relationship with morphology over large evolutionary time frames, which makes it much more challenging to use these data sets for reconstructing specific morphological structures in ancestors. Given they have such a broad range of functions in different phyla, our ability to use them as proxies of morphology is very challenging. That's what we've argued anyway. So back to the question is, what is this data good for? So if it's not particularly good for reconstructing ancestral morphologies, what use does it have in terms of a comparative data set? And what I think for these comparisons is that there is a very useful role for these, particularly in animals that have very divergent body plans, that no one makes head nor tail of the morphology. So no one has a problem making a comparison between a head here and a head here, a thorax and abdomen. The basic organization of an arthropod and a vertebrate is relatively straightforward because the morphologies look pretty similar. But that's much more challenging. Trying to make the comparison between this animal and that animal is much more challenging. And what we've argued is that these data, while they don't tell us whether or not these structures are homologous, anatomically homologous, what they tell you is there's regional similarities that clearly these different anatomies have evolved from. So it suggests that the forebrain and the proboscis evolved from a common ancestral structure. We don't know what it looked like, but it suggests that these are as regions, homologous regions, that have followed very different morphological trajectories depending on the ecological pressures that animals have experienced along those different lineages. Similarly, the midbrain and the collar evolved from a structure that we don't know what it looked like, but it allows these basic comparisons between these very morphologically divergent body plans. So if your goal is to try and understand in an ancestral deuterostome whether there's any morphological homology, you can at least use these gene expression domains as maps to tell you, okay, if I'm going to find some sort of morphological homology between somewhere in the collar and somewhere in the midbrain, then we have to compare these two regions of the animal. We can't compare the proboscis to the midbrain. We can't compare the trunk to the midbrain. We use these maps as a way of digging deeper into the morphological comparisons of the different animal groups. So this approach we've used more recently with a project which I think is the most challenging one, which is how to make head nor tail of the echinoderm. So what the hell is an echinoderm adult? How can you make any sense of the zoology of this animal? And Libby Hyman, in a sort of invertebrate treatise, she's pretty opinionated about lots of things, but I really like this quote: "I salute the echinoderms as a noble group especially designed to puzzle the zoologist." And I think that holds true today as it did back in 1955. And this is a project, strangely NASA was really excited about funding this project, which I would be very excited about, but it does make some sense. If you're interested in why they funded it, I can give you the story. So echinoderms are really fascinating because they start off life as a perfectly respectable bilaterian. So this is a starfish larva that shows an anterior and a posterior. Here's the mouth, the ventral side opening up, and it has a dorsal side. So as a larva, they're very simple animals, but they clearly have a dorsoventral axis and an anteroposterior axis. I think they're beautiful animals. Somewhere in the middle of larval development, they start to form their adult body plan on the very posterior around the stomach. So this is actually a baby starfish that you can see grafted onto the posterior part of the larva. So this will actually become, at metamorphosis, the actual baby starfish here. So in the genome of the starfish, there is a set of coordinates that define not only a perfectly respectable bilateral axis, but it also codes for this rather unusual body plan. So can we use the same approach that we used in Hemichordates, using this conserved suite of body plan genes to try and understand how to make any basic comparison between the organization of the radial body plan of the adult echinoderm to that of an adult chordate or adult hemichordate? And over the years, there's been a whole range of different hypotheses about echinoderms. So if you think of a starfish here, so many people have argued that the mouth to anus axis is the ancestral axis, and that each of the rays of a starfish are more analogous to a limb than they are to a primary apiaxis, whereas other people have argued that each ray of a starfish replicates a duplicated axis that has been duplicated several times. So a starfish is actually 5 apiaxes joined at the head. And so there's very specific predictions with that. And given, if we believe that these genes mark apiaxes pretty well, then they may give us some clues to how the body plan of the echinoderms is organized. And that's kind of a big roll-up to say that, so this is more sort of formulated. So we know now from our work in chordates and hemichordates that the ancestor of hemichordates and echinoderms must have had an apiaxis, and it must have had the same suite of genes involved in specifying that axis. We don't know what it looked like, but we know its apiaxis defined by the same suite of genes. So what happened on that lineage leading to the adult echinoderms? Are we going to find this cryptic similarities by looking at this suite of genes? Or did the whole thing get blown apart and reassembled into completely novel topologies, which tells nothing about the organization of the echinoderms? So we just started to look at this. And so 6-3, which is this anterior forebrain marker and proboscis marker in hemichordates. So if you look at the starfish after metamorphosis, so we're actually looking at the starfish turned upside down. So we're looking at the oral surface and each of the underlying ambulacra. So the nervous system runs along each of the ambulacra in 5 different chords. So this is the mouth, and these are the ambulacra extending with 2 feet on either side of the ambulacra. So this tissue forms nervous system as the animal starts to develop. So this entire surface is being stained by this gene 6-3. NK2.1 is overlapping in expression with 6-3, but is not expressed up into the 2 feet. And then this more posterior gene, OTX, is actually not expressed in this central nervous system domain. As you can see, this is not stained, but is expressed around the 2 feet and in the ectoderm and mesoderm on either side. So we don't know what this means yet. So we've only got 3 genes, and these are actually pretty tricky in situs to carry out. But I think it's starting to tell us there may be something to the idea that the region that's homologous to the anterior may be the entire oral surface of the echinoderm. That's what may be formed from an ancestor that had a much more complicated nervous system and a more traditional bilateral organization. So the other thing that we're interested in more diversity is that the work we've done on hemichordates has really focused on this one species, and this species is a bit unusual in the sense that it's been great for functional studies because we can knock out genes and they form an adult in a period of about 3 or 4 days. So you can manipulate adult development by introducing things into the egg right after fertilization. However, the most common form of development in Hemichordates and in Echinoderms, which is their sister group, is larval development. So this is a direct developer. Most Hemichordates have this kind of development. So they go through an extended period in the plankton, and these are beautiful larvae, and you can actually find them off the coast here at the right time of year. They swim around for up to 2 months in the plankton, and their larval body plan is completely unrecognizable from their adult body plan. So there's an extended period of larval development, up to 2 or 3 months, and then only towards metamorphosis does the adult body plan start to form. So there's an uncoupling between larval and adult body plans. So one question, and this is work that my grad student Paul Gonzalez has really been working on, and this is a species that he's been working on from Morro Bay. So one hypothesis is that if we look at this larval developing species, we'll find the exact same findings that we found in Saccoglossus, that in early development you'll start to see the major regions of the body plan are starting to form. So you'll have an anterior domain, a middle domain, and a posterior domain, which means essentially that the larva has a very similar organization to the adult: anterior, middle, and trunk. If you look at the classical literature, however, in larval morphology, so Thurston Nakali in 2005 published this really nice little paper where he has argued that Deuterostome larvae are actually head larvae. So they don't have a trunk, that they basically are just swimming heads. And so only at metamorphosis do you get the formation of the trunk. So in his scenario, we'd get something like this, where all the anterior genes would be expressed normally, and the middle ones would be there, but we get no trunk expression until metamorphosis. So Paul had very specific predictions for what he'd find. So he's looked at a range of different genes, but I'll just show you a couple of markers of proboscis, collar, and trunk from Saccoglossus. So this is this species, Schizocardium californicum. And what he found is that the anterior genes, as Thurston Nakali predicted, seem to be expressed relatively normally. So we get anterior restriction during larval development. And then you can see here that basically by late larvae and early juvenile development, after metamorphosis, looks essentially the same as Saccoglossus. So it looks like the anterior genes expressed in a pretty similar way to what we see in the direct developer. And similarly found that the mid genes, such as PAC6 here, are expressed not in the anterior, but more in the middle region of the embryo. And then essentially at metamorphosis, they look identical. LIM15 is a particularly good example. This is a really nice marker gene that shows the sort of middle region of the embryo. And basically between the hemichordate and the direct developer and the indirect developer look identical after metamorphosis. And he also found, for a couple of posterior genes, a very similar expression pattern. So this gene HOX910 is a very posterior HOX gene, and like we see in Saccoglossus, it's expressed in the most posterior ectoderm. So initially he was very disappointed because he wanted to look different. But then when we looked at all of the HOX genes, we found that most of them are not expressed during larval development. So this is just a quantitative PCR at different time points of development showing anterior genes. So this gene 6-3 I showed you as a sort of forebrain marker and a proboscis marker. And PAC6 and OTX are both expressed down into the sort of middle regions. So these different time points, 10 hours, so these are the embryonic stages of the larval body plans forming. So the first 4 time points are basically larval body plan, and the last 3 time points are basically adult body plan. And you can see that basically 6-3 gets turned on at gastrula, and it seems to be important in the formation of the larva, and then goes up through the roof during metamorphosis. PAC6 and OTX the same, that only at these very early stages, at 10 hours after fertilization, you see no expression, but then you start to see activation of the gene in early larval development and all the way through into metamorphosis. However, if we look at the HOX genes, you can see in these early time points, HOX1 is really not detectable in the early larval stages. It's only during late larval development, adult development, that they start to be activated. And that's the same for HOX1, HOX3, HOX4, HOX5, HOX7 does seem to come on earlier than the other ones. HOX1113 and HOX1113, again, seem to be all delayed until metamorphosis. HOX910, like HOX7, is expressed early, but the majority of these trunk-defining genes are not activated until after the formation of the larva, and you start to begin the formation of the adult. So Thurston seems to be pretty on the money with his description from the morphology, that if you look at the larva and you compare that to direct developer, you get a very, very different story. So you have to be very careful about making generalization about phyla for one particular developmental strategy. In order to make broad comparisons about the development of a particular phylum, you have to sample the diversity of the different life histories, larval or direct developers. And I think I'm going to stop because I have I ended up spending more time going through some of the basics, but this gets into a much more developmental story, which I think is really, we've run out of time for. So I'm going to skip through this and go to my acknowledgements. Boy, I really am. So, the work that I showed you was Paul did the work on the indirect developing species, and Ari, who left my lab a little while ago, did all the work that I was going to show you. And then in terms of helping the lab, Kevin has been my lab tech for the past 6 years since I've been here, and has been an invaluable help for the larval rearing and the natural history. And then the work has been supported by NASA, by the Brain Research Foundation at University of Chicago, and then also by NSF Developmental Mechanisms. So we have a little bit of time for questions if I haven't completely lost everyone during that description.
Regions of the body, reasons for the dye to show itself. You were just injecting it into RNA?
No, so what we do is you fix the embryos with formaldehyde, just a basic fix with a buffer. You then clone the genes that you're interested in, and then we put them into a plasmid, transcribe RNA, messenger RNA, which has one of the bases has been substituted with something that basically has a modified base with a little marker on it. So you then take that probe and you bathe the embryos that have been fixed with that probe. So that probe basically diffuses it into the embryo. Wherever it finds the complement to that gene, wherever that gene is actually expressed in the animal, so this is actually an antisense copy of the sense messenger RNA. Right, so normally you produce sense RNA that's read by the ribosomes, right? We make the reverse complement of that, which is complementary, so that when it finds an RNA copy in a cell that it's identical to, it will bind and form a complex to that. You can then rinse all the other probe out, but when it finds a match, it will bind it. So any cell that's expressing that gene, that probe will stick to. And then using antibodies, we can then recognize those epitopes we've added to the probe, which will then precipitate the dye. So it's a basic, if you know immunocytochemistry, just looking at using antibodies to recognize different neural epitopes or whatever, it's a similar idea, but you're recognizing RNA rather than protein. Yes?
And the other question was, so the development of those embryos was on like you said a couple day scale, and I know fruit flies are used a lot for
yes.
like quick generational studies and stuff like that. Has anybody, and you might have mentioned a little bit of it, but has there been any work where to look at ecological stressors determining how things are I guess, evolutionarily, if anything changes? Using fruit flies on that short
So using how much the environment affects development, basically. So that's something that people are just really getting interested in. So development is a complete antithesis to ecology in the sense that we try and keep everything as constant as possible, right? So we don't want any kind of variation. In fact, you'll never see a statistic in a developmental biology paper for the most part because you really sort of select what is sort of standard and obvious. People are realizing that when they've been doing these mutation analyses, so if you try to pick apart a gene and figure out how a gene is turned on and off, and there's a whole battery of enhancers and inhibitors that activate a certain gene, they found that when they actually knock out many of these enhancers, there's no effect on the animal whatsoever at their laboratory conditions. And now people are realizing that if you start raising those knockout animals that seem to have no phenotypic effect in different environmental temperatures or under ethanol stress or in a variable environment, suddenly they start to see phenotypic effects. So people are suddenly starting to use ecology to try and understand how this might influence development. But people are only just starting to do that. For the most part, people ignore environment and they want to keep things standard as possible. But it's a great question. Yeah?
Is there a reason for the trunk genes not turning on? Why wait till the metamorphosis and when you're turning into an adult to turn on the trunk gene?
So if you think about what trunk is about, right? Trunk is largely sort of locomotory. In all the locomotion in larvae is really by cilia in the anterior. So maybe they just don't need a trunk as a swimming larva, and the trunk really is only important for benthic burrowing. But in the plankton, it's more of a hindrance. It's just going to kind of weigh you down. That's just total waving my arms. But what's important about those data are that typically when people make developmental comparisons across groups, they look at embryos. And if you pick a species that has a larva and you're making comparisons to other phyla that don't, you're going to be looking at a completely different genetic network when you're making generalizations about a phylum. So all of the major groups that we look at are direct developers. So arthropods and chordates are all direct developers. So Hox genes come on right away. Echinoderms, hemichordates, most phyla you can think about have a much more extended larval period. So if you were only to sample a larva, you would come to a very different embryological conclusion than looking at direct developer as well. So the broad comparisons across phyla are very sensitive to the life history strategy you pick as your representative species. Yeah, of course.
Why is NASA interesting?
So they have this exobiology program, which is sort of based on trying to understand life on other planets. So if we were to try and find life on Mars, then what would we look for? What are the criteria we use for looking for it? And so most of what they fund is sort of early life decisions. So assembly of macromolecules, the origin of multicellularity, and how cells are self-assembled. There is one line in their program that says that they're interested in assembly of more complex life forms, and the angle that I use for this project, which I actually kind of like, is if you are to understand the evolution of life on another planet, you need to understand axis formation, because as soon as you get more than a couple of cells, you need to specialize. And in order to specialize cells, you need to organize them along an axis. And we understand so little about diversity of axis formation. Echinoderms are an example of a group that have completely broken the rules of axis formation. And so understanding how axis formation has changed in echinoderms might give us some basic principles about how axes can change over time and how we recognize an axis. That was my pitch.